Hi, I'm Tyler Browne from Western University. I'm going to talk to you about my work on developing a generalizable Cas9 sgRNA prediction model using deep learning and transfer learning techniques with small, high-quality datasets. A big question in genetic engineering is what region of DNA you should target. Specifically, what is a good sgRNA target site sequence, and how can we tell? Backing up a little bit, CRISPR-Cas9 is a genetic engineering tool with broad applications for gene knockouts, gene editing, and potentially as a novel antimicrobial. The Cas9 nuclease is targeted to a DNA site by a guide RNA, which we will refer to as the sgRNA. There exist a large number of potential sites for nuclease like SP-Cas9 at a rate of about 1 site per every 16 nucleotides. We know that variations in the sequence that is targeted have large impacts on nuclease activity. So the question remains, how do we choose the best site? There do exist a few Cas9 sgRNA prediction models. However, most of them are developed for eukaryotes and generally perform no better than a random guess when it comes to prokaryotic predictions. Prokaryotic models do exist. However, there are few, and there's a decent amount of room for predictive improvement. Here we present a new deep learning model using a novel neural network architecture and transfer learning to predict bacterial sgRNA sequence-associated Cas9 activity for the nucleases TEV-SP-Cas9 and SP-Cas9. Our data comes from two sources: pre-existing bacterial data generated by GLBIO et al., and our two new sgRNA activity datasets, where sgRNA activity is measured by the cleavage of a toxic plasmid by either TEV-SP-Cas9 or SP-Cas9 in E. coli. These datasets are used to construct a deep learning model containing a dual branch architecture with both convolutional and recurrent neural network layers. The existing larger ESP-Cas9 dataset from GLBIO et al. Was used to train what we refer to as the base model, which you can see in the left mid figure. With transfer learning, we use the parameter weights from the base model as the starting point for training our TEV-SP-Cas9 and SP-Cas9 models. To prevent overfitting of the model during transfer learning, we froze a portion of the layers, including all convolutional neural network layers in the model. Moving to the center, one note is that the performance metric that we use is the Spearman correlation coefficient, which reports a statistic measuring the rank order of model predicted activities for each sgRNA versus their experimentally determined activity rank. As we can see in the top middle figure, following transfer learning, both nuclease versions of our model display improved performance over the predictions of all prior models, with our TEV-SP-Cas9 model reaching a rank correlation of 0.63 under 5-fold cross-validation. As we can see, the 5-fold cross-validation results of our TEV-SP-Cas9 and SP-Cas9 models perform quite well, and you can see that in the bottom middle figures, A and C. To test generalization and ensure that our models are not overfitting on our relatively small datasets, we tested both models on an E. coli SP-Cas9 dataset of about 7,800 unique sgRNA sequences. Both versions of our model performed well on this unique dataset, as you can see below in the middle figures B and D, with our TEV-SP-Cas model, figure B, obtaining a rank correlation of 0.68, which is a substantial improvement. Moving to the right, to obtain optimal performance, we tested expanding the target site sequence seen by the model. We added nucleotides either upstream or downstream of the target site to the model input and tested the effects of this on predictive ability, as we can see in the rightmost figure. We found that the inclusion of 8 nucleotides downstream of the sgRNA target resulted in the maximum performance from the model, indicating that non-target site nucleotides do have an impact on performance and an impact on activity, in line with prior experimental results. Unlike downstream nucleotides, the inclusion of any additional upstream nucleotides resulted in a decrease in performance. Overall, we are presenting CRISPR-Hal an accurate sgRNA prediction deep learning model, which demonstrates the benefit of transfer learning with small, high-quality datasets and exceeds the performance of all prior models on TEV-SP-Cas9 and SP-Cas9 nuclease sgRNA-associated activity prediction. Right now, we are in the process of generating a larger sgRNA activity dataset in the species Citrobacter rodentia, which will be used to develop a robust cross-species bacterial Cas9 sgRNA activity prediction model. As of right now, our model, CRISPR-How, is available for public use through GitHub. Thank you for listening to my talk. I appreciate the interest. Thank you.